Augustine the fruit fly was flying around one day with his fruit fly friends, eating fruit and swimming. His life was extremely short, a couple days at most, so he is trying his best to enjoy his incredibly short and pathetic existence. Augustine always dreamed of doing big things. However, he had such short time to do it, so what was the fly to do? He was feeling pretty down on his luck and was kind of thinking of just giving up. But then he remembered something. His colony was being plagued with a genetic illness which led to some flies being born with small, non-functional, or no wings. These vestigial wings led to them having an even shorter and more pathetic existence. Augustine and his best friend Bartholomew decided to survey the colony to figure out the cause of this mysterious illness so that they could do at least something of value while they were around. Vestigial wings are caused by major deletions or insertions of thousands of base pairs in the vestigial gene on the second chromosome of the fruit fly's 3. Both parents pass down an allele, and the recessive lowercase v allele creates the mutation. If the child gets one dominant allele and one recessive allele, the mutation does not happen. If they have both recessive mutated alleles, then the VG protein is either misfolded or not produced. This protein helps scalloped proteins regulate cell death, and without it, mass apoptosis occurs in the wings during development. When the fruit fly grows up, these wings will either be slightly weirdly shaped, be like half the size of normal, or non-existent, depending on the type of mutation in the allele. In most cases, it is fatal. Augustine and Bartholomew, instead of acquiring elementary biological knowledge, decided to fully research, develop, patent, and manufacture a magical shrink ray in a matter of hours in order to shrink themselves down to the size of an amino acid chain. They then entered the nucleus of a developing larvae in order to see what caused such mutations. Because both parents of the larvae had a long history of perfectly healthy flies, they knew that this would be a normal, healthy one. After locating the vestigial gene on the second chromosome, the flies saw a normal gene thousands and thousands of base pairs long. The messy string-like formation was being translated by mRNA, using helicase to unzip the DNA and RNA polymerase to attach complementary nitrogenous bases to the long, single-stranded mRNA. They then followed the mRNA to a ribosome outside the nucleus and saw the mRNA zip through the ribosome. Each 3-letter sequence or codon coded a complementary anticodon, and these anticodons were embedded into the tRNA, which then coded for a specific amino acid to be produced by the end of the tRNA. The flies watched in fascination as thousands and thousands of amino acids were formed, joined by peptide bonds. These amino acids were then formed into the vestigial protein, and the flies followed it and realized its main use was regulating cell death in the development of the wings with the help of the scalloped proteins. After the flies returned to normal size, they saw the larvae hatch into a happy, healthy baby fly. Afterwards, they then went into the child of 2 parents which had a history of deformities. And this larvae specifically had the mutated alleles, both of them, and so had a mutated gene. What they saw was intriguing. When they went back inside the nucleus and onto the vestigial gene, they saw a massive insertion of over 8,000 random base pairs on a certain part of the gene specifically called VGBG. They knew what this would cause. The mRNA transcribed everything, including the false insertion. The mRNA then translated all the base pairs through the ribosome to create amino acids. But because the base pairs were all screwed up, so were the amino acids. I think you can see where this is going. As for the type of mutation specifically, in this case, it produces many different amino acids that differ from what is originally intended in this specific case. So in this instance, it would be a missense. The flies observed that the VG protein produced by these mutated amino acids was completely dysfunctional and misfolded, and so could not help the scalloped proteins regulate the cell cycle in the wings during development. And so a good amount of the cells died, and so As a result, the fly was born with very small wings, and this led to its eventual demise. After concluding that a mutation in genetics was the cause of the disease, it was only through mass surveys and tests that Augustine and Bartholomew were able to realize the allele aspect of the problem. With this new information, they informed the colony, and after a bit of careful breeding, the mutation was eventually gone from the colony. Augustine and Bartholomew were heroes. The end.